Hello guys, my name is Sana and today we're going to talk about cloning. So, when you clone, you produce a genetic cell that is identical to its donor. There are different types of cloning: DNA cloning or gene cloning. There exists a parent organism whose fragment of DNA is taken to a bacterial plasmid where it can generate itself. Then the cloned DNA splits in the new host cell. Reproductive cloning. First, the DNA of a cell is extracted and placed in an egg. The original nucleus from the egg is removed so the cell can grow into an embryo. It is planted into a mother through artificial insemination. Finally, when the embryo is born, his DNA is identical to his parent. Therapeutic cloning is used for researcher purposes and consists of cloning of the human embryo. First, a nucleus is taken out of human egg cell. It's replaced with a nucleus from one of the patient's cells. Then the egg is triggered to develop into an embryo. 3 days later, that's how it looks. 5 days after, cells are removed from the embryos with a pipette. Then, stem cells from an embryo are grown in a dish containing nutrients. Finally, stem cells develop into different tissues and organisms. They can be used for medical treatments. Benefits of cloning: it modifies faulty genes to incorporate the corrected genes to the host. It can genetically modify food, adding nutrients or plants in order to be better or to be resistant and look out for insects. For species survival, there are endangered species which may benefit from cloning since it has immense potential in a rising population. Human cloning. The stem cells can produce tissues to be used for medical treatments as cancer, Parkinson, or Alzheimer. Risks of cloning. There are 1 to 10% of the attempts to clone an offspring that are successful. The costs are quite expensive for that little effectiveness rate, including all the losses. Mutation is a risky option, and 4% of the cells cloned in animals have had abnormal infections. The greatest risk in human cloning is the unpredictability of the development of intelligence in these clones. Also is a concern the possibility of the clones being treated differently from others. After all of this, we know how cloning works, but what did humans do to get to these conclusions? In 1962, Sir John Gurdon answered a question that had been hanging over cell biologists since before the turn of the century. Are the cells of an adult organism genetically identical to the fertilized egg from which they are derived? Briggs and King in the early 50s suggest that cell genetic material is irreversibly altered as they begin to differentiate. But Gurdon showed that this was not the case. By transferring the nucleus of an adult frog cell into enunciated eggs, he obtained cloned adult frogs. He isolated and cultured intestinal cells of frogs of the species of Xenopus leavis. Then, he isolated unfertilized eggs of the same species by irradiating them with ultraviolet light to destroy their nucleus. Subsequently, he isolated the nucleus of the intestinal cells and injected into the coralless eggs. After a few weeks of development, he got new tadpoles. Dolly was born in 1998, in July 5 in the Roslin Institute, Middleton in Scotland. She was cloned by Keith Campbell, Ian Wilmot, and colleagues at the institute. She had 3 mothers. One provided an egg, another the DNA, and the third carried the cloned embryo. She was created using the technique of somatic cell nuclear transfer, where the cell nucleus from an adult cell is transferred into an unfertilized developing egg that has had its cell nucleus removed. The hybrid cell is then stimulated to divide by an electric shock, and when it develops, it is implanted in a surrogated mother. She died at the age of 6 because of a lung disease and severe arthritis. Then we have Promethea, which is the first cloned horse and the first one to be born from and carried by its cloning mother. She was born in the Laboratory of Reproductive Technology in Cremona, Italy. Dr. Cesare Galli and others at the lab experimented with 841 reconstructed embryos. Of the 14 available embryos, 4 were implanted in surrogate mothers, only that Promethea was the success in being born. Moving to the opinions and ethics side, as you can see, many religions are opposed to every kind of cloning. 2 of them are restricted, and just realism allows cloning. The UNESCO says in its 11th article, reproductive cloning of human beings is a deep violation of human rights and dignity. As a conclusion, cloning has some serious consequences. Animal cloning usually causes congenital malformations, premature aging, blindness, deafness, diabetes, and low efficiency. As a clear example, Dolly the sheep, with 270 attempts. On the other hand, we have human cloning, which causes moral consequences since a human cannot be killed just to create another humans, and lots of embryos are produced to obtain the cells and destroyed eventually, also because of the waste of money and time. Knowing this, what do you think about cloning? This is all. I hope you enjoyed it. Thanks for watching.